Welcome to the ORG.one product demo. My name is Dan Fordham, and I'm the lead for the ORG.one project. In case you didn't know, ORG.one is Oxford Nanopore launching a project to try and address the biodiversity crisis. So we offer free kits and consumables to Oxford Nanopore customers that have access to critically endangered species, and on the proviso that they upload them within 6 weeks of receipt. So, through the provision of those free of charge kits and consumables, we aim to reduce the barrier of entry and create a powerful and open and equitable network. We realize that not all researchers have access to the same resources, so we continue to look for methods to reconcile this. For example, one of the requirements is for the user to upload the data within 6 weeks of receipt of the flow cells. This does keep the project fresh and keeps it moving, but it also allows those without bioinformatics capacity to upload the data, and those with the expertise to be able to take the data, process it in an expert manner, and then share it back with the users. And we have a Slack channel that we've put in place for this kind of networking. So if you want to join it, please come and contact me, and you can come and join in. So like I said, we probably have 3 types of users: those with samples, those with sequencing capacity, and those with bioinformatics capacity. And any one user can have 1, 2, 3 of those. So we aim to try and join those people together, and so in this demo, we're trying to demonstrate that. So we've already loaded this library to demonstrate that sometimes some samples need greater sequencing capacity, and with the P2 Solo that we're using here today, we're aiming to deliver this. Now, we're running 2 true ORG.one samples today. We're going to try and sequence and assemble them, and one of those is the large copper butterfly, and I have my friend here, Ben Price from Natural History Museum, who's kindly delivered the sample. Yep. Ben, would you like to introduce yourself?
Yeah, sure. Thanks, Dan. I'm Principal Curator at the Natural History Museum here in London, and I'm responsible for partly responsible for around 30 million insect specimens that we have in our collections, going back around 200 years, and more recently looking at how we can extract genomic data from those collections.
Oh, thanks. Could you give us a brief overview and background of the sample that we're sequencing here today?
Yep. So we're sequencing here today the large copper butterfly, which is a species of butterfly that went extinct in the UK in the 1850s, and there were some reintroductions in the early 1900s, but they failed. So we're looking at sequencing its genome and comparing it to some of the European populations.
And why is it of interest? Why is it of particular interest?
Well, it's of interest because of this local extinction event, and because there are these 3 subspecies. And so we know nothing about the genomic data of these species or subspecies, and we know nothing about the historical population sizes of the species in the UK before it became extinct. So by generating this reference genome, we can compare our UK specimens in the Natural History Museum that date back to before the extinction event, and we can compare it to the populations in Europe, and that will help inform conservation of the species going forward.
That's really interesting.
Thank you.
So how useful is the genomic data in this context?
Well, we can't do any of that sort of population genomics and comparative genomics with our collection without a reference genome. And so to build that reference genome, we need fresh material, and so this genome will go a long way to allow us to compare the historical material we have in our collection. So it's critical. And so
this is very interesting. And so what we find with ORG.one is that sometimes we're able to produce genomic data, but users don't necessarily know what to do with it. So what do you think are the barriers to the adoption of genomic data for conservation purposes?
Yeah, that's a really good point. I think we need a lot more collaboration and communication between conservation organizations and genomic scientists and genomic data providers, as it were. So it's fantastic that you have the Slack channel where anyone can kind of network and help with that process, because conservation scientists might not necessarily have the bioinformatic capacity. So I think it's really just about communication, about showing through examples like this that this technology exists and what we can do with this technology so that others can take it on board.
That's really great. Do you have any other projects you'd like to tell us about?
Oh, yeah. There's a lot of different genomic projects at the Natural History Museum. I think 2 that sort of stand out are at both ends of the sort of read length spectrum. So we've got the Darwin Tree of Life project, which is aiming to build reference libraries for all 70,000 species in the UK. And then at the really short end of the spectrum, we're looking to genome skim our up to 250-year-old specimens to fill gaps in reference libraries for global DNA discovery, essentially.
Did you say how long this has been extinct in the UK?
So this the species went extinct in the UK in 1851, I believe.
Okay. And it's gone missing from fenland areas, is that right?
Yeah. So the hypothesis behind its extinction was that fenlands were drained for agriculture, and this resulted in its host plant dying out, and so as the species went extinct.
And are you pleased how well the sample is sequencing today?
It's amazing. So we've got here 23 gigs, which is, what was it, about 50x coverage, and we started this 9 hours ago, which is incredible.
Excellent. So, thanks very much. So all data will be made available. It will all be made available through through ENA. So as part of the program, we will continue to upload the data, and we upload the raw data, so people are able to develop tools and rebase call if they want to, and it's available to everyone. So if you want that, please get in contact with me. We're also running another sample today, which is the rollaway monkey, which has come from the University of Nottingham, and in collaboration with Twycross Zoo as well. And so that's been running along at the same time, and that too has produced around 23 gigs. So what we're showing here is within about 48 hours, we've gone from sample through to what we hope is still going to be a draft genome assembly, and showing the power of the P2 and a deployable sequencer delivering the high throughput sequencing almost everywhere in the world. So thank you.